Pilot is an interpretable machine learning model for the analysis of multi-scale spatial and single-cell data across samples. The main aim is to find disease trajectories. This allows us to find patients at early stage of the disease and to find potential molecular markers for diagnosis. Pilot can also be used to find potential subgroups of patients and find molecular mechanisms associated with these groups. This could be explored in personalized treatments. Deep molecular profiling techniques are multi-scale. That is, the data measured for one patient is represented by a distribution of cells, and each cell is at a distinct molecular state and has a particular cell type. To estimate how similar these multi-scale molecular profiles of two patients are, we explore a mathematical approach named optimal transport. Moreover, we use nonlinear regression models to characterize molecular and cellular changes driving disease trajectories or subgroups, which allow the interpretations of results. We also use Pilot to analyze complex clinical relevant deep molecular profiling data. By the analysis of myocardial infarction spatial atlas, we can recapitulate cellular and molecular changes related to tissue remodeling in patients with ischemia. We also apply Pilot to digital pathology data where we shown that Pilot trajectories are predictive of patients at risk of kidney failure. We observe an increasing use of spatial profiling and digital pathology techniques in tissue biopsies and their application in large disease cohorts. Pilots represent a unique framework to dissect unknown patterns related to the disease progressions. We are currently working on kidney and heart and blood diseases as a cohort, where Pilot provide a useful insight in the detection of patients at risk. Deep molecular profiling techniques are expensive and might not make it into clinics. However, we are able now to do spatial molecular profiling in the same samples where histology is available. This opens the opportunity to use deep learning algorithms to learn how to read molecular features from histology, which is a standard used in biopsies of samples. We are currently performing research on how to predict molecular signatures from standard histologies, which will measure molecular markers in histology in an expensive and flexible manner, improving both diagnosis and treatment guidance. It is essential to consider the complexity of biological systems and analytical measures using measurements. Understanding your data and doing quality control is crucial to obtain meaningful results. For example, we observed in a study with blood samples of COVID-infected patients that Pilot could group patients by city of measurements. This indicates that deep molecular profiling data might have experimental artifacts that mask real biological signals. Unfortunately, we observed that this is still ignored in some scientific studies. The project was mainly funded by DFG via the Clinical Research Unit 5011 as well as System Medicine Consortium Fibromap, financed by German Ministry of Education and Science.